Right. So welcome everyone to the ICTS Colloquium. It's a pleasure to have you all attend, at least virtually. Today's speaker is Vinothan Manoharan. He is the Wagner Family Professor of Chemical Engineering and Professor of Physics at Harvard University. He received his PhD from the University of California, Santa Barbara in 2004 and worked as a postdoctoral researcher at the University of Pennsylvania before coming to Harvard in 2005, where he's been since. Vinny's research focuses on understanding how particles in a liquid, say a suspension, how they organize themselves into structures such as crystals and viruses and also living tissues. His lab uses optical microscopy and holography to visualize this self-assembly in real time. And their goal is to discover new physical principles that underlie these kinds of complex systems and to apply them to practical problems in a variety of sciences, medicine and nanotechnology, amongst others. So viruses, of course, have been part of the collective conversation for more than a year. And in today's talk, titled The Physics of Virus Assembly, Self-Assembly, Vinny's going to give us some insight into how these viruses are formed, as well as some of the interesting physics that goes on with that. So with that, I'll hand it over to Vinny and invite him to give the talk.
Okay, thank you very much, Vishal, and thank you everybody for joining this afternoon. I want to invite you all to ask questions during the talk. So if you do have a question, I don't know what the protocol is, but I can see everybody. So you can raise your hand virtually or just speak up however you need to be, however you like. So the work I will tell you about was primarily done by a former postdoc in my group, Reese Garman, who's now a faculty member at San Diego State University, and a former student, Aaron Goldfein, who's now working at the National Institutes of Science and Technology. And several other current students in my group are involved, Linnell Williams, Tim Chang, and Amelia Payne. So I want to, I will give some background on viruses, and I recognize that in today's day and age that many of you are already experts on viruses, but I felt it was probably good to just give some basic concepts in case anybody was not familiar with what viruses are. So the viruses, the way I describe them is that they are containers for genes. And what I'm showing here, these are different reconstructions, usually from electron microscopy, of different types of viruses, some of which you are probably familiar with: HIV, influenza, HSV. These are all viruses that infect humans, and you have heard of before. HK97 is another one. Others, infect different organisms. CCMV and TMV are viruses that infect plants. MVM is a virus that infects mice. Lambda is a virus that infects bacteria. But what, these viruses, they come in different shapes and different sizes. Certainly, that's what you're seeing here. And they infect different hosts, but they have in common that they all are containers for some nucleic acids, either DNA or RNA, and that is their genetic payload. So the virus that I'm going to talk about today is one of the smaller viruses. It's a so-called simple RNA virus. It's similar to these viruses over here. It is called MS2, and this is the virus we work in my group that we work with. It is a virus that infects bacteria. It infects E. coli bacteria, which is a bacteria that we all have inside our gut, in our intestines. And so all of you likely, and me too, we all have some of this virus inside us because it is infecting the E. coli that are in our gut. And what I'm showing here, this is a reconstruction from electron microscopy of the virus structure. What you're seeing here are the proteins that make up the shell or the capsid of this virus. And it's difficult to see from this reconstruction, but If I'm to draw this schematically, what it would look like is you would have 180 or 178 in this case copies of the coat protein. That's the same coat protein, surrounding on the interior a long piece of RNA of about 3,600 bases. So, for reference, that's enough. That's enough RNA to encode about 4 proteins. So this movie, which hopefully will play and you can see, you can, as this rotates around this projection, you can see that the virus actually has some nice symmetry, which I'll talk about soon. And then in a moment, you'll actually be able to see from this reconstruction the interior of the virus. This is just a lower resolution reconstruction at the moment. And then now we see the interior, and then we see the RNA in there. You can see that the RNA from this image is actually pretty well structured. And that is because RNA is a polymer that can bind to itself, and it can form this secondary structure, which I tried to draw schematically here, with these kinds of places where it's folded onto itself and it makes these loops and hairpins. So how does such a virus work? I'm going to show you not MS2 for the moment, but a similar simple RNA virus that infects humans, which is poliovirus. Poliovirus is still in some areas a threat to human health, but by and large, the vaccination campaigns have really helped in sort of mitigating that threat to human health in a lot of areas. But poliovirus is similar size and shape to the MS2 virus that I just talked about. So for those of you who are familiar with viruses, and for those of you who are biologists, forgive the very elementary explanation here, but I thought this would be useful because often those of us who come from physics and engineering and math aren't necessarily familiar with the central dogma of molecular biology. The central dogma is that information in cells is stored in DNA. That DNA is then transcribed to RNA, which acts as a messenger molecule, and that RNA is then translated to protein by ribosomes inside the cell. And so the way that an RNA virus, a simple RNA virus like poliovirus or MS2 works, is that they kind of short circuit this, this procedure. In other words, the RNA from the virus gets directly translated to protein. So what happens is that the virus, this diagram may be a little bit too hard to see, so I can make some bigger labels here. The virus first attaches to the host cell, and the means of attachment depends on the virus, but it attaches in such a way that it can release the RNA into the host cell. The host cell is then, the ribosomes inside the host cell then see that RNA and think it's just an RNA that occurs in the host cell and start translating that directly to protein. In these simple RNA viruses, the first protein that is synthesized is actually a protein that copies the viral RNA. It's called a replicase protein. And so the host cell starts translating the RNA into this replicase protein. That protein can then make copies of the RNA. Those copies get translated into, again, get more copies of the replicase. And so you get an amplification of the viral RNA in the cell. And that RNA also encodes for many other proteins. In particular, it codes for the coat protein of the virus. And what will then happen is that the cell basically fills up with the RNA from the virus and the coat protein. And then a rather remarkable thing happens in this soup of protein and RNA. Self-assembly occurs where new viruses are formed. The proteins self-assemble around the viral RNA. Those new viruses break out of the cell, usually by breaking and destroying the cell, killing the cell, and then go on to infect other cells. So that is the basic life cycle of an RNA virus. It's the simplest kind of life cycle in that the RNA gets directly translated to protein. There are more complex viruses, retroviruses, DNA viruses, where there are additional steps involved. This is the simplest mechanism. Just pause here to see if there are any questions. Okay. Now, the question on all of your minds is, what does coronavirus do?
Maybe I'll ask a question about your previous slide. So you said that the first proteins it synthesizes are replicas of the original. So how does it know that? I mean, what determines the sequence? Or are you going to talk about that? Or
that's a great question. So I'm not going to talk about that, but there are a few ways that it's a very good question, because it's just a piece of RNA, and so how does the virus tell the cell to do this first? And it has different ways of doing this. So the RNA, first of all, the RNA has a secondary structure, so parts of it are folded, and that means that parts of it are inaccessible to the ribosome. And there's another very interesting way in which MS2 in particular can encode which proteins are synthesized, and that is that two proteins can actually be encoded in the same stretch of RNA by a so-called frame shift. So normally we know that a codon for an amino acid consists of 3 nucleotides. But if you shift that over by 1 nucleotide, you can also get a string of 3 nucleotides, but it's a different string of 3 nucleotides. And so that frame shift allows the virus to control, because it's control the order in which proteins are produced, because there's only a small probability that the ribosome will attach in the wrong frame. So the virus has some various, if I'm allowed to anthropomorphize the virus, it has various clever ways of controlling which proteins occur first.
Okay, thanks.
Okay, so coronavirus is on all of our minds, and so there are some similarities between how the SARS-CoV-2 virus works, which is the virus that causes COVID-19, and these simple RNA viruses. Both of them work in a similar way in that the RNA in the virus, they're both RNA viruses, and the RNA in the virus gets directly translated to protein when it gets into the host cell. In the coronavirus, though, the coronavirus structure is much more complex than what we see in these simple RNA viruses, and they're also much larger, and I've tried to draw things to scale here. In the RNA virus, the simple RNA viruses that I'll talk about, the capsid, the protein shell, is roughly spherical. It actually has icosahedral symmetry, and the RNA is about 3,000 bases, which means it's a small number of genes. Coronavirus, the capsid is helical, so it's not this same icosahedral shape, but it's surrounded by a lipid envelope, a lipid bilayer, which is roughly spherical and which actually comes from the cells that it infects. And at the surface of that bilayer are these spike proteins, which give the famous corona in the images that you see of these viruses in the electron microscopy images that we've all seen. And those spike proteins are the target, essentially, for the immune response that vaccines seek to create. So this is a much larger and more complex structure than what we see in our simple RNA viruses. But in a similar fashion, the coronavirus is will release their RNA into the host cell, and that RNA gets translated to protein directly by the ribosomes. I'm going to limit myself to these icosahedral small RNA viruses in this talk, and I'm not at all an expert on coronaviruses, so I won't try to extrapolate what we've learned to coronaviruses, but I think there's a lot of interesting physics just in these, even though these things are quite simple. There's a lot of interesting physics in these small RNA viruses. And in fact, this is why we started studying viruses. I mean, my background is in engineering and in physics, and my interest was always in self-assembly and statistical physics. And viruses are very interesting from that point of view. I think those of us who come from outside of biology and medicine area would always like to have some impact in that area of like doing something that will help people who are afflicted by viruses. But of course, we have to keep in mind that we're working a bit far from the real human health challenges that are out there. And so what we learn may not always be directly useful to those challenges. But in the long run, we hope that this kind of fundamental research may have some impact there. Also, it's just worth keeping in mind that especially today, I think we all have a very we all have an antagonistic relationship with viruses, in that we know the costs in human lives of viruses that infect humans. But it's worth keeping in mind that viruses infect every single organism on Earth. And that means that they actually have a very beneficial role for us in many cases. The viruses like MS2 that are in your gut play an important role of regulating the bacterial ecosystem that is essential for how we digest food, for example. The microbiome. And the viruses that are in the environment actually play an important role in the carbon cycle as well, because they infect bacteria, that cyanobacteria, which do a lot of photosynthesis. So viruses are important to all of life on Earth. And that means that they're interesting quite broadly. And also, from a physics perspective, the thing that got us interested in viruses is the fact that they self-assemble, and these small RNA viruses at least self-assemble. And so, what do I mean by that? For these simple RNA viruses, you can take the capsid protein, put it in a test tube, and the viral RNA, put it in a test tube. And these are purified components. There's nothing from the host cell here. You can mix those two together, and they will spontaneously form virus-like particles with the shape and size of the virus. And in some viruses, in fact, when you do this process, you can actually get an infectious virus at the end. So to some people, that's a scary thing, because it seems that like you can synthesize viruses in the lab. But this is only true for a few, this is only true for these simple RNA viruses. But it's still a remarkable thing. If you look at the viruses that are assembled in vitro, they look almost identical to the wild type viruses. And remarkable, I say, because it's very hard to make a synthetic system that does something like this. It's very hard to make a synthetic system that will spontaneously organize itself into such a structure, unassisted by any dissipative mechanisms, any energy-consuming mechanisms, any direction, essentially, from the cell. So we have a question, will self-assembly still happen if the RNA used is not the viral RNA? Great question. So I will come to some experiments on that towards the end of my talk. With certain viruses, like the plant viruses, in fact, you can get them to assemble around almost any charged polymer. It doesn't even have to be a nucleic acid, could be just a charged polyelectrolyte, like a polymer that has some charges along the backbone, or even charged nanoparticles as well. And so those viruses are sometimes used for lots of materials applications and gene delivery applications and drug delivery applications. MS2, it seems to be a little bit more specific to its own RNA, but the reason for that is not clear yet. And it's an interesting question in all of virology, because the host cell is filled with all sorts of RNAs, and a question is, when a virus gets in there and starts producing its protein, how does it specifically assemble around its own RNA as opposed to all the other RNA that's in the host cell? And this is an open question in the field. Yeah. Okay, so good question. Please keep the questions coming. So today, I think many of you may have heard of this term self-assembly, and it's used quite broadly in the fields of nanotechnology and chemistry and nanoscience. But it actually was originally coined to describe the process by which viruses form. And it was originally coined by Casper and Klug in this famous paper, which I'll talk about physical principles of viruses. And they were contrasting this process of the proteins coming together to form the shell with, for example, how the proteins themselves are produced. The proteins themselves are produced by a ribosome, which attaches amino acids one by one to a growing chain. So that's a directed process. Whereas the virus shell is produced by all the proteins spontaneously coming together. And so they called that a self-assembly process. And the thing that we should keep in mind is that at the scale at which these things are happening, everything is Brownian. Everything is basically stochastic. The motion of all of these proteins and RNA is stochastic. So what I'm showing here is just a simulation from Brownian dynamics done by Mike Hagen's group at Brandeis that tries to show how a virus capsid might assemble from some subunits, and a very simple virus capsid from some subunits, and the RNA, which is shown here as this red charged polymer. And I show this only to say that we don't know if this is how it happens, but this is just to show that we expect that at this scale, everything is stochastic. Things are randomly coming together and falling apart and randomly coming together again. And what's remarkable is that even though that is a stochastic process, it leads to a non-trivial structure. In general, a very high yield. This process exceeds 50% yield, 70% yield, something like that, of functional nanoscale virus particles. So why is it that we get this kind of symmetry, this kind of structure? This is a question that goes back a long way, actually, to Crick and Watson's other famous paper, not the most famous paper by Crick and Watson, but the other paper that they published in Nature. They asked the question, what sort of structures viruses might have? And this was in the early days of structural biology on viruses. Only a few viruses had been studied by X-ray methods, and nobody had yet looked at the viruses in high resolution and electron microscopy. But Crick and Watson made a prediction. What they argued is that a virus has to, a virus is a small thing. The nucleic acid could really only encode for a limited amount of, it really only can contain a limited amount of information. So they argued that on the principle of genetic economy, that is, the genetic material is limited, that viruses need to be efficient in how they use that genetic, that amount of genetic information. And so they said that, okay, what is the simplest way that you could then make a shell which encapsulates this RNA? And they argued that, well, you would want something with a high volume to surface area made from many small proteins, because you have a limited amount of information there. And in particular, you want to, the virus, again, if I'm allowed to anthropomorphize, wants to reuse the same contacts between proteins, because those, if you have to make a complex protein that has many different ways in which it can bond to other proteins, then that requires more genetic information. And so this is, they predicted that one of the possibilities was that the virus would arrange into an icosahedral structure. So this is a diagram of an icosahedron. The geometry, for those of you who have not seen this in a while, so an icosahedron is a platonic solid, 12 vertices, 20 regular triangular faces, very high symmetry, as with all of the platonic solids. That prediction actually was borne out by later electron microscopy studies. And then we have a question: are there other platonic solid structures in viruses? This is an interesting question. So I think 50% of known viruses are icosahedral. There are other shapes that come out in the virus. There's a lot of tubular viruses, cylindrical viruses. And as with all things in nature, you always have exceptions to rules. And so there are viruses with other kinds of shapes as well. But icosahedral symmetry seems to be the most prominent type of symmetry. And there's an elegant kind of mathematical reason for that, which I'll talk about here. So here's a question to ponder for a moment. Say you have an icosahedral structure like this, and you have proteins. Proteins in general are not symmetric. They have no axis of symmetry, no planes of symmetry either. So how would you then arrange such proteins on a symmetric structure such that you don't have to create complex, you don't have to create a lot of different ways in which they can bond to each other? An equivalent way of saying that question is, how would you arrange such proteins on the structure of an icosahedron such that they are all in symmetrically equivalent positions, meaning you can rotate any one of them into any other? And probably many of you already can see what the solution is. You take each of the triangles, each of the triangular faces on the virus here, and put 3 proteins on each triangular face. And if you do this, you can hopefully see that you can then rotate any of the proteins into any of the other. They're all in exactly symmetrical positions. And we call this the T=1 structure. There are 60 proteins, 3 per face. And indeed, many viruses actually do have this structure. So that sort of validates the idea, that observation. But it turns out that there are icosahedral viruses with more than 60 proteins. And Casper and Klug tried to figure out, well, what would the structure of these things look like? Because mathematically, 60 proteins is the largest number of points. That 60 is the largest number of points you can put on a sphere and still maintain symmetry. So when you go beyond that, you have to break symmetry in some way. And so Casper and Klug said, is there a way to break symmetry that would minimize the energy cost of breaking symmetry? And they came up with this clever construction. They said, well, imagine you put the virus proteins on a lattice, on a triangular lattice, and then you draw your triangular facet on this lattice. And there's various ways of drawing the triangular facet. These are all shown here. And they can be described by these 2 integers, h and k, which describe the movements along the lattice vectors. And if you can then cut out, they actually did this by hand. They printed these or drew these on paper and then cut out different configurations, and then you can fold them up into a icosahedral shell. And what you see in this case is that some of the proteins are in different sites than others. So here we see that, it'll be more clear on the next diagram. Some of the proteins in this T=3 structure. So this is the first one I showed you where they're all in equivalent positions. In this structure, however, some of the proteins are now in these pentagonal regions and some are in these hexagonal regions. And so they occupy symmetrically distinct positions. But Casper and Klug argued that the strain involved here, which is the energetic cost, would be fairly small because you just have to scrunch these guys up a little bit. And in return, you can still form this shell. And they went through the math and said, well, if this argument is correct, then viruses should come in certain, icosahedral viruses should come in certain integer multiples of 60 proteins. And they gave the integer sequence. And the integer sequence was 1, 3, 4, 7, and so on. And these are the structures that they predicted for that integer sequence. And indeed, this is correct. The icosahedral viruses do follow this integer sequence. So, there are viruses with 60 proteins, and then the next size up is 180, and then the next size up is 240, and then you keep going. And at some point, you have to break the mirror symmetry, and they have to become chiral. So that's a beautiful example of mathematics and physics explaining the structure of a biological system. But for our purposes, I think this is quite interesting for another reason, which is it raises the question of how does that structure form? So in the virus that we're talking about, all of these proteins are the same protein, right? But yet that same protein, it's the same protein chemically, but yet that same protein has to arrange itself in different ways. It has to arrange itself in different local configurations in hexagonal and pentagonal sites. And how does it do that is the question. So another way to look at the structure of the viruses that we're talking about is as it looks like the old-fashioned soccer ball. I think you probably call this a football. Where you have these pentagonal facets and hexagonal facets, and we can think about this in the language of condensed matter and say that some of the proteins have to form topological defects in the virus. That's required by the spherical symmetry, by the topology of the sphere. And how does it put those defects in the correct places? That's the question, when all it really knows are the local interactions between proteins. So that's the question we hope to answer. And experiments have tried to look at this question of how does the virus form? But they've given only really indirect information on that. I won't go into detail in these experiments, but an issue is that by and large, these experiments are bulk experiments. That is, they look at many viruses forming at once. And our goal when we started this project about 6 years ago was to actually look at an individual virus as it self-assembled. And so these are the group members who contributed to this. So viruses have been very, very well studied in biology, and in particular in this field of structural biology. A tremendous amount is known about the structure of viruses since Casper and Klug's paper. This electron microscopy image that I'm showing is an example of the power of modern structural biology. You can tell from these images where every single atom is of every single protein in the virus with atomic scale resolution, with something like 2 angstrom resolution. It's a remarkable degree of structural information that you get. But what you can't get from these kinds of techniques, at least not yet, is anything about dynamics, about how these structures form. The temporal resolution is very, very limited. And so our approach was to do something in reverse, which was to say, can we get information about the dynamics or kinetics of how the structure forms, while still not necessarily being able to tell everything about what the structure is. And so we decided to do this optically, to use light to measure those dynamics. And the immediate challenge that you can imagine is that the structure I told you about is 28 nanometers in size, whereas the wavelength of light is about 500 nanometers. And so I noticed there's a question in the chat. Question was, Do they image single viruses, or do they have to form crystals to image them? Yeah, good question. So the traditional approach is to look at crystals and then do X-ray diffraction, but the reconstruction I'm showing here was done by electron microscopy, and it's actually a so-called asymmetric reconstruction. So they average over a small number of viruses to be able, or small groups, viruses to be able to determine the structure. So there's usually some ensemble averaging involved here. Single viruses can be imaged, but the amount of, you can't get to the structural resolution that's shown here without doing some averaging. And that's important because, what we want to do is look at individual viruses and get some idea of what the heterogeneity is in the way that they form. So yeah, so light, how are we going to do this with light? This thing is smaller than the resolution limit. So we're going to give up on getting any structural information from this system. And then, but the bigger issue here is timescales. Simulations and experiments have, before we did our experiments, had limited the timescale to between microseconds and minutes. I'll give you a pause just to think about that for a moment, right? So that's 9 orders of magnitude, just to be clear. That's or 6 orders of magnitude. It's an enormous amount, 9 orders of magnitude, right? And that means that we need an instrument with a large dynamic range. So how do we do this? Well, what we do is we use elastic scattering to actually look at a virus. We shine light on the virus as it's growing, and we measure the intensity. Now, Rayleigh taught us that you can do this, but the scattering is very, very weak. The scattering scales as the 6th power of the particle size, which means it's a very, very weak signal for small things. However, the advantage here is that you can do this without damaging the virus. It's non-invasive. So we do a little trick, which is an old trick from Gabor, which is that you take the scattered light and you interfere it with a reference beam. And by doing so, you get an interference pattern. But if you work out what the intensity should do, well, it's the sum of the field squared in this case. And so your signal, you can ignore in this case the scattered intensity because it's so weak, but your signal consists of a background term and a mixing term, the interference between the reference and the scattered light. And that mixing term scales as the radius cubed instead of the radius to the 6th power. So it's still not great, but it's a much better signal than you get from just scattering alone. And the price you pay for that is some background term. So there's a very clever way of reducing the background, and that was done in this apparatus that was designed by Philip Kukura and Vahid Sandogdar, called the interferometric scattering microscope. You shine light back in through a microscope objective, through the back of the microscope objective. And then say you have a virus that's sitting in water above a cover slip. What happens is that the light will transmit through the cover slip, but a little bit will get reflected off the cover slip, a little tiny amount. That's your reference beam. And then that reference beam interferes with the light that is scattered from the virus. And the net result of that is you get a signal which is proportional to the mass or number of proteins that you have in this region. So that's the important thing to remember about all the data that I'm going to show you. The intensity that I will show you, just think of it as a proxy for the mass or the number of proteins in the thing that's assembling here. So here's I'm going to show you some images now just to show you that we can see individual viruses using light. So this is fully assembled viruses. They attach onto the cover slip, and this is the movie hopefully showing, yes. Each of those dark spots, that's a virus that just plopped down, a fully formed virus that just plopped down onto the cover slip. And these are individual viruses that you are seeing using light. And you can see that in all cases, the size of these spots is diffraction limited, right? We're still limited by diffraction here, but you can detect those small spots. And the signal to noise you get here, the intensity, you can see here the intensity jump is quite large compared to the noise here, which is shot noise. So how we watch individual viruses assemble is that we basically we put the viral RNA, we tether the viral RNA onto the surface, and then we flow in protein, and we watch these spots corresponding to the RNA to see how the intensity changes over time. And the intensity should change over time because the mass is increasing, number of proteins is increasing, and so the scattering is increasing over time. Okay, so I'm going to show you the movie, which shows how the virus assembles. And so this is a wide field technique. So we're seeing an area of about 10 microns by 10 microns. What you see now is just noise. It's a very sensitive technique. We're really pushing the limit of sensitivity here, though. And so you'll always get background noise. So there's nothing that you're seeing here. This is just random noise. I'm going to start the movie. You'll see some spots over here. That wave was when we injected the protein. There's some spots here which are just junk. Ignore these two things. And what you're going to start seeing is some dark spots forming. Okay. And you'll see they'll form at different places and at different times. Each one of those dark spots, I'm going to argue, represents an individual virus particle forming and assembling on an RNA that is tethered to the cover slip. And this movie is sped up by about a factor of 5, but we're recording at about 1,000 frames per second, so 1 millisecond temporal resolution, but over a minute of time. So we're dynamic ranges, 6 orders of magnitude in this experiment. So what information can we get from this? Well, what you can do then is to look at each of these spots as they form. And you can look at the intensity as a function of time. And it looks something like this. Starts at a low level and then jumps up. And so this is the point at which we added protein. Now, remember, intensity in this case is sort of a proxy for number of proteins or mass. And what we were able to do is do the same experiment on a nanoparticle, just to show that you do end up with these full capsid shells. That's what's being shown here. These things are the full capsid. And we could calibrate the intensity of a full capsid. We know what intensity corresponds to a full capsid. And so what that is to say is that this intensity that we see this spot jump up to, that corresponds to roughly the size of a full capsid. So we argue that, in fact, that this trace that I'm showing here represents the assembly of an individual capsid almost to completion. We can do that again with multiple spots, and we get different curves that I'm showing here. And what you'll notice is that these traces seem to take off or rise at different times. This is all the data for about 60 different viruses. Each of these traces represents the assembly of an individual virus. And if we plot the number of assembling particles as a function of time, what we see is it follows this curve. And remarkably, that is fit extremely well by an exponential function. So what does that tell us? Well, that tells us that there is likely some barrier, energy barrier, free energy barrier to the growth of the virus. And another way of saying this is that this kind of kinetics that we've observed is consistent with a single-step nucleation and growth process. So Gibbs developed the idea of classical nucleation theory. The idea is very simple, that as a structure starts forming, he thought of it in the context of, say, a crystal or a liquid. But as that condensed phase starts forming, you have the surface energy dominates. And so the total energy goes up. But at some point, the bulk energy starts favoring, the bulk difference in chemical potential starts favoring the formation of a larger thing. And so in our virus, we imagine that there's a barrier to nucleation corresponding to a critical nucleus, which consists of a few proteins that then can grow to completion. And in this case, yes?
Vinny, there's a question in the chat.
Oh, Do you also see disassembly after the full assembly? No. The virus is, in this case, is thermodynamically stable. So it doesn't disassemble. So you have to do a trigger to get that to disassemble. And so we can do that by introducing some different change in conditions for the virus. But it's an open question as to how disassembly proceeds, and it generally proceeds in different ways depending on the virus. Thank you for that question. So everything we've seen is consistent with nucleation and growth, that there seems to be a barrier. And another question is, do you expect the glass surface to affect assembly? Yeah, another good question. So it's possible. However, what I can say is that the viruses look to be complete. So there are a number of viruses that can assemble around a tethered piece of nucleic acid, and it seems that it's possible for that nucleic acid to just snake through the proteins. And the virus is fairly far away because we use a long tether from the glass surface itself. However, there is a diffusive effect. Diffusion can be affected by the presence of that surface, so that does affect the kinetics in the system.
Vinny, just before you move on, sorry, I had another question. You showed these plots where sort of the intensity suddenly rose up.
Yes.
Is that an experimental effect, or is that an actual thing? Is that because the threshold suddenly went through your experiment, or is that how the assembly happens?
Yeah, this is how it happens. Yeah, this is a good question. So what's going on here? So let's look at one of these traces. So in the context of nucleation and growth, right, if that's the correct model, then these traces you can interpret as follows. So you add the protein here, and then you have to wait a while before a critical nucleus forms, and then a critical nucleus forms, and this is the growth, and then it just starts growing the shell on that critical nucleus, and then it reaches completion. And so in the context of that theory, what that's telling us is that growth happens very rapidly after the formation of a critical nucleus. And also we notice that it doesn't go up and go down again, which means that the critical nucleus is probably quite small and comparable to the noise level that we're seeing here. And both of those are important points for the conclusions that I'm going to draw.
So as a follow-up question, you mentioned that the current literature with respect to how long self-assembly took was about 9 orders of magnitude. So are you saying that it's actually much quicker?
In a sense, yes, but the range is the literature for that 9 orders of magnitude was that the difficulty is that with any nucleation and growth process, you have to wait a long time, usually for nucleation. In this case, we have to wait minutes for nucleation, but then once nucleation happens, the process occurs in seconds. And so with such processes, you expect a wide range of timescales over which things might occur because you have these two processes of induction or nucleus formation followed by growth. And so that, I think, is why it was difficult to pin down the time range. But it may be quite different in other viruses. I should point that out as well.
Right. There's another question from Shashi in the chat as well. There seem to be very few steps in the time trace. Do these correspond to single protein assembly events? Yeah,
great question. So this, I think you may be referring to some traces like this, and we believe that these correspond to double nucleation events. So in that case, what may be happening here is that you have a virus starts growing, and then another nucleation event happens, and another shell starts growing. And we see that that happens more often at higher concentration, and we're actually able to get some images of structures at these higher concentrations. And what you can see is they look like a bunch of partially formed viruses that have come together, and this is consistent with what you would expect for multiple nucleation events. I think that's what you may have been talking about, Shashi, from the multiple steps. And then the other kinds of small things here are we think, just noise, these fluctuations. Okay, so the big picture here is that it appears that the RNA, this is a heterogeneous nucleation process, and the RNA sets up, in a sense, a competition between nucleation and growth. And it depends on the protein concentration. And as you decrease the protein concentration, you reduce the nucleation time. And so what that ends up happening is that when the nucleation time becomes comparable to the growth time, you start getting these malformed structures. But at the very least, we have good amount of evidence that the nucleation is that this is a nucleation and growth process. And another question, is it true that the RNA is essential for nucleation to start? I would say that the RNA reduces the nucleation barrier. It is possible to get, in many of these systems, empty capsids that will nucleate, but you either have to wait a long time for that to happen, or you have to change the conditions, the solution conditions for it to happen. So the RNA reduces the nucleation barrier. And this is a case where you might extrapolate what we've learned to some interesting health implications. So if you wanted a virus to be less infectious, one way to do that might be a little bit counterintuitively to increase the rate at which it grows. If you increase the nucleation rate, you increase the probability that the virus is going to form a malformed structure. And that's actually a strategy that some research groups have been looking at into designing viral therapeutics, is actually to increase the virus rate of protein production. Okay, so I am running out of time, so I will just end here. I think there's an interesting, I started with this interesting question of, does the nucleation and growth explain how the virus forms this complex structure? And in fact, our experiments have opened more questions than I think they have raised, that they have solved. That in particular, what we found is the apparent critical nucleus size is small. It's only about 6 proteins. And so how do the proteins know, after you form a small cluster of 6 proteins, that then now you have to start forming pentagons, and you have to start forming those pentagons in exactly the right places to get the right curvature and the fully closed structure? We have no idea how that happens. And also, it seems that once that nucleus forms, growth proceeds monotonically, so there doesn't seem to be any barrier to forming that beautiful icosahedral structure. And even those malformed structures have the right curvature. So somehow the growth is determined at the formation of the critical nucleus. And it's not clear how that happens, but our current the thing we're looking at is what role the RNA plays in that assembly. And I won't go too much into detail here, but a brief description of our results is that we find that changing the RNA does not affect the yield, but does affect the nucleation rate. So how that helps the virus assemble, we don't know yet. But I'll stop there and take a few more questions. Just want to conclude by saying that there's a lot you can learn from these individual virus measurements. For example, you can determine the growth mechanisms, but what remains to be understood, as I've said, is how is that assembly process cell wall controlled? That question that I started with and introduced is still an open question. So with that, I want to thank all our collaborators and our funders from the National Science Foundation. And I really want to thank all of you, and thanks to Vishal and Rajesh for inviting me, and thanks to all of you for your questions and your time.
All right, great. Thank you so much, Vinny, for that wonderful talk. I'm going to open the floor to anyone. If you have any questions, just go ahead and unmute your microphone and ask the question, or if you feel more comfortable, throw it into the chat. Maybe I'll use my prerogative as the chair of the session to ask a question. So you said, it's still an open question as to how the topological defects are introduced in the self-assembly process. It's my naive guess that the experimental technique you're using is not going to be able to resolve that question. And so you would need something that can resolve the structure, possibly at the level of electron microscopy, as well as have the kinetics at the same time. Is that a reasonable assumption?
That would be the ideal case. It seems, however, that that would be the ideal case. It seems, however, that such techniques are a long way off. There are advances in electron microscopy that give time-resolved images, but I think that's a long way off. Our approach is going to be actually to combine to simulation with experiment. So we work with Mike Hagan's group at Brandeis, and this is a role where computation and theory can be essential. So we get highly quantitative data from these experiments. If you can make a model, theoretical, simulation, computational, that is able to reproduce those characteristics, the quantitative data that you see in the experiment, then you may have a sense of how the structure forms that you can then test in some way using the structural biology techniques that are available to you. So I think this is a case where you can really do a lot by combining theory, computation, and experiment.
I see. There's a question in the chat. If the RNA regions responsible for assembly can be identified, then maybe blocking that region by something like siRNA might stop viral assembly. Is it naive to think so?
It's not at all naive to think so. And with MS2 at least, our results have shown that, and we've blocked those regions by using small pieces of DNA, and it doesn't seem to affect the yield of assembly, but it does affect the kinetics. So you reduce the nucleation rate. And so other viruses, though, it may be quite different, right? So I don't know what it's going to be for coronavirus, for example. But this is certainly a strategy that some for human infectious viruses that some people are pursuing. So it's not at all naive to think that it's a good idea.
Vishal, can I ask a question?
Yeah, go ahead, man.
Yeah.
Go ahead.
Lovely talk here. Very nice. I enjoyed it. So in the simulations that Mike Hagan does, he sort of builds these pentagons and hexagons, and these are what attached to each other to form these icosahedral. But that's very different from your experiments. Of course, you start off with the proteins itself. So is it true that they initially form these pentagonal plates and then come together, or is it a direct assembly of proteins that? I mean, does it form? Does the entire capsid form via first forming the pentagonal plates and then them attaching to each other?
Right. No, that is a good question. I didn't talk about this, but the experiment suggests that it's individual proteins that attach to the RNA first, and then they come together on the RNA. So quite different from the scenario that I showed you in the simulations. Some degree of coarse graining is, of course, necessary in all the simulations. And so I showed you the very coarse-grained ones where it's basically a T=1 structure forming from 12 pentagons. But an interesting thing from that I've learned from talking to Mike is that it's very hard actually to make 180 proteins come together in a simulation into the right structure, which also serves to just show that the problem is non-trivial of how the virus is able to figure out what the right interactions are to form the T=3 structure. It's hard to get those correct interactions in simulation.
Maybe a follow-up. Can you explain? Sorry. Go ahead, Rajesh.
I mean, I just had a quick question. Probably you just answered it, but were you saying that under certain circumstances you could, even in your in the way you prepared these viruses, you could get the T=1 structure as opposed to the T=3? I mean, is that what you were saying? Is that something that you can actually do in the lab by changing some concentrations or whatever, go from between the different structures?
Oh yes, this is a very interesting question. Most viruses, it's hard to get them to form a different structure. So like you could take a very small piece of RNA, for example, and the same protein, and you might think that the virus would form a smaller shell around it. But in fact, that's not usually what happens. Usually the curvature remains the same. It just packages more of the small RNAs. So it seems to be quite hard to get it to avoid what the the structure that it prefers, T equals 3 in this case. But with MS2, there's been some work by Danielle Tolman-Ursek's group at Northwestern where they make a single amino acid mutant, and in some cases that can lead the virus to form actually a smaller capsid. And there are other viruses where you can change the size of the capsid by also making similar mutations. But it's an interesting question is like why does it so strongly prefer this this particular curvature?
Vinny, there's a question in the chat.
Yeah, so a question about can the X-ray measurements in synchrotron be used to make movies of such viral self-assembly? I don't know. I actually don't know what the state of the art in this area is. Probably some people in the audience may have a better idea than I do about this, about what the temporal resolution is now. I think it'll be hard with most of these techniques to get to the kind of temporal resolution that we have in the optical experiments. But again, the advantage that they have is they get far more information about structure.
Are there any other questions from the audience? Oh, just one in the chat.
What is the smallest object you can detect? Oh, yes, good, yeah. So Philippe Kukura's group has shown that you can actually detect single proteins optically, but there's a trade-off between the size of the object you can detect and the averaging time there, because you're limited by the number of photons that you get. And so if you want to detect single proteins, your temporal resolution is limited to on the order of seconds. Whereas if you want to detect, in our case, our sensitivity is 6 proteins and our temporal resolution is sub-second. So there's always that trade-off, and that's not something you can easily get around. So if we want to be able to see both the nucleation process happen, we want to get that long dynamic range, but also see the growth which occurs over a small time scale, then we have to limit our sensitivity to a few proteins, about 6 proteins in our experiments. So that's the trade-off. But the question of how many monomers make a nucleus is exactly what we want to answer somehow. We'll just have to do it indirectly.
Any other questions?
Just again, a random question, Vinny. I know you don't really think, I mean, you haven't really worked on the coronaviruses, but they also probably have a discrete symmetry and not just a spherical symmetry. Is that very naive? I mean, because after all, they have some discrete spacing of all these corona spikes. So is it one of the the non-convex polyhedra symmetries?
I think so. Again, I'm not an expert on coronavirus very much at all, but I think for a lot of these enveloped viruses, where the outer shell is actually a lipid bilayer rather than a protein capsid, that there's not a lot of symmetry there. I think that bilayer is basically disordered, from what I understand. However, I do understand that in coronaviruses there is a self-assembly step that takes place at some point in the immature virus. So the proteins do have to come together around the RNA, but I don't know if that involves a highly symmetric structure at that point.
Thanks, great talk.
Thank you very much, and thank you all for the questions, and especially want to thank the, as I said, NSF and also the Simons Center, and I know that ICTS also has a Simons Center as well. So looking forward to more interactions between our two centers.
Yeah, definitely. We should be in touch, and hopefully, once the situation gets better, we can actually do some joint activities.
So thanks, Vinny, for that wonderful talk. Thanks to everyone else for attending this as well. And Vinny, thanks to all of us virtually. We would be clapping in person. So it was a great talk. Thanks again.
All right. Thank you all, and hope to see you all in person soon.
Bye, everyone.
Bye.